I want you to just close your eyes for a second. Imagine you're standing in a cave.
But not just any cave.
Right, exactly. This is the Altai Mountains, southern Siberia. It is this rugged, honestly breathtaking landscape. You've got steep cliffs, these endless boreal forests, and winters that are so cold they can literally freeze the moisture in your breath the second you exhale.
It's brutal outside.
But you are inside Denisova Cave. It's this massive limestone cathedral set high up on a cliff face, just overlooking the Anui River.
And it's the kind of place that it just feels ancient when you stand in it. It has this perfect, stable microclimate, cool and dry, which, as we are going to see today, is really the hero of this whole story.
It really is. So the year is 2008. You're an archaeologist, and you're working in the dirt. It is very unglamorous work. You're basically sifting through sediment that is just packed with the debris of thousands and thousands of years.
Hyena poop, bare bones, tiny stone flakes.
Yeah, all of that. And you are looking for human remains. But you aren't finding skulls. You are not finding skeletons laid out in these beautiful burial rituals. You're finding fragments. Just these tiny, shattered splinters of bone.
It's basically the ultimate needle in a haystack, except the haystack is made of other needles, because most of those bone fragments are completely unidentifiable. They just get labeled as mammal and put in a bag.
And then you spot something. It's a tiny piece of bone, specifically a distal phalanx.
The very tip of a pinky finger.
Right. It is so small, maybe the size of a pea or a really small pebble, you could so easily flick it away without even noticing it was there. It does not look like much at all.
No, it definitely doesn't look like the key to completely rewriting human history.
And yet, that single, non-diagnostic little chip of bone,
Cataloged as Denisova 3.
ended up being the biological equivalent of a supernova. It just blew everything up.
It really did.
We are doing a deep dive today into the Denisovans, and I have to say right up front, if you think you know this story...
Like if you read a headline a few years ago about cavemen in Siberia.
Exactly. If you think you know it, you need to wipe the slate clean. Because the research that has come out recently, specifically between 2024 and 2026, has completely overturned the old narrative.
It has. We really used to think of them as just a footnote. You know, a weird, obscure cousin of the Neanderthals that we just didn't have much data on.
But the new picture...
The new picture is wild. They were a massive, highly sophisticated, hyper-diverse population. They controlled a huge swath of the globe for hundreds of thousands of years.
And the twist here, the really big twist that we are going to build up to today, is that they might actually be more closely related to us, to modern humans.
More than Neanderthals are, yes, which is a massive claim.
It changes everything about how we view our own family tree.
Completely.
So our mission for this deep dive is to give you a long, totally understandable explanation of this mystery. We are going to look at the chemistry, the fossils, the geography, the whole legacy. But I think we have to start with that ghost status.
The molecular evolution.
Right, because unlike Lucy or the Neanderthals or Homo erectus, we didn't find the Denisovans by looking at their bones first.
That's the craziest part. Usually, paleoanthropology is intensely visual. You find a skull in the dirt, you measure the brow ridge, you look at the angle of the jaw, you say, okay, this is a new species.
But with Denisovans...
We had the code before we had the container.
So take us back to that moment. It's 2010. That little pinky bone from Siberia has been sent off to the Max Planck Institute in Germany. Svante Pääbo and Johannes Krauss are the lead geneticists there, and they decide to extract mitochondrial DNA first. Why start there? Like, why not just go for the whole nuclear genome immediately?
Well, it comes down to preservation. Think of a human cell like a giant library. The nuclear genome, the main DNA that makes you who you are, is the central archive in that library.
Okay.
It's massive. It has 3 billion letters. But there are only 2 copies of that archive in every single cell. One from mom, one from dad. So if that specific book gets damaged over 50,000 years, it's just gone.
You can't read it.
Right. But mitochondrial DNA, or mtDNA, is totally different. It lives outside the nucleus, in the mitochondria, which are the little power plants of the cell.
Right. I remember that from high school biology.
The powerhouse of the cell. Exactly. It's a much smaller book, only about 16,500 letters. But the crucial thing here is that there are thousands of copies of that smaller book in every single cell.
Oh, I see. So if you're working with a bone that's been sitting in freezing dirt for millennia, the odds of finding at least one intact copy of the mtDNA are just way higher.
Exactly. It's the low-hanging fruit of ancient DNA. So they sequenced it. And they were probably expecting it to just be a Neanderthal.
Yeah.
Because we knew Neanderthals lived in that same cave.
Or maybe an early modern human.
Sure, that was possible too.
Mm.
But instead, the sequencing machine spit out something that made absolutely no sense.
It was a complete shocker.
A total shocker. They compared the sequence to modern humans. No match. They compared it to Neanderthals. No match. The genetic distance was huge.
So it was a distinct lineage.
A third player on the board of human evolution that literally nobody knew existed until that exact moment.
And this is where the name comes from, right? Because they didn't have a skull, they couldn't actually give it a formal Latin name.
Right. Taxonomy has rules. To name a species officially, you usually need a holotype, a physical diagnostic specimen that defines what the species looks like. You have to be able to point to it and say, this specific shape of jaw and this slope of the forehead defines the species.
And you just can't do that with a pinky tip.
You really can't. So they just called them Denisovans after the cave where the bone was found.
It's funny because Denisovans sounds so casual compared to Homo sapiens or Homo neanderthalensis, but it stuck.
It did. But to prove this was actually a new group and not just some weird mitochondrial fluke, they needed the nuclear genome.
Which brings us to section 1 of our deep dive, the molecular detective story. Because getting that nuclear genome turned into this high-stakes detective novel involving chemistry and supercomputers.
It's honestly a miracle we have this data. We really have to appreciate how difficult it is to sequence ancient DNA, or aDNA.
Because DNA isn't meant to last forever.
No, not at all. When an organism dies, the biological repair mechanisms that keep your DNA intact while you're alive just stop working.
So the DNA just starts falling apart.
It fragments. It breaks into these tiny, tiny pieces, usually fewer than 50 base pairs long. So imagine trying to read a massive novel that has been put through a paper shredder.
That sounds impossible.
It gets worse, because the letters on those shredded pieces of paper actually change over time.
Wait, the letters change? Like typos introduced by time?
That is exactly what it is. It's a chemical process called deamination. Basically, over thousands of years, the DNA-based cytosine, the letter C, loses an aramine group and turns into uracil, the letter U.
But uracil isn't even supposed to be in DNA, right? Isn't that an RNA thing?
You know your biology.
Yeah.
Yes, uracil is usually an RNA. But when the sequencing machine tries to read that corrupted DNA fragment, it sees the uracil and it just reads it as a thymine, a T.
Oh, wow. So everywhere there used to be a C, the machine sees a P.
Right. So the text isn't just shredded, it's actively corrupted. The word cat becomes bat or tat. If you just read the raw data from the machine, you get gibberish. It's just a biological error code.
So how did they fix it?
Paavo's team had to develop incredibly sophisticated computational frameworks. They built software that could actually predict these error patterns.
Like an autocorrect for ancient DNA?
Sort of, yeah. The software essentially looks at the data and says, okay, I see a T at the end of this fragment. But based on the specific damage profile of this 50,000-year-old bone, that T is actually the ghost of a C.
They reverse engineered the rot.
That is a perfect way to phrase it.
Yeah.
They mathematically reconstructed the original sequence, and they managed to get what we call 1.9-fold coverage of the Denisovan nuclear genome.
Meaning they read every single letter of that girl's code almost twice, on average.
Which is incredibly high quality for something this old. And that nuclear genome confirmed it. The Denisovans were a sister group to Neanderthals, but they had been on their own separate evolutionary path for hundreds of thousands of years.
Okay, so we have the ghost. We know they exist. We have their code. But we still have this massive problem that haunted the field for an entire decade. We still do not know what they look like.
We were building a puzzle, but we didn't have the picture on the box.
Right. And you can't just keep hoping you'll stumble across more pinky bones with perfect DNA. You need a better way to hunt. Which brings us to this revolutionary technique called zooms.
ZoomS, zooarchaeology by mass spectrometry.
I love this part because it shows how archaeology is basically turning into high-tech forensics.
It really is.
Yeah.
So here's the problem ZoomS solves. Excavations at places like Denisova Cave generate tons of bone waste. Just thousands and thousands of tiny bone shards that are way too small to identify by their shape.
Like you can't tell if it's a deer or a human or a wolf.
Exactly.
Yeah.
And historically, these were just tossed in a bag labeled indeterminates and shoved in a museum basement.
Because you can't run DNA extraction on all of them. It's too expensive, it takes too long, and it destroys the sample.
Right. But you can run zooms. ZoomS doesn't look at DNA at all. It looks at collagen.
The protein.
Yes, collagen is the main structural protein in bone. It is incredibly tough. It survives way, way longer than DNA does. And crucially, The exact amino acid sequence of collagen differs just slightly between different species.
So a cow's collagen fingerprint looks different from a hyena's.
And a hyena's looks different from a human's.
Hmm.
So researchers started screening these thousands of trash bones from the cave. They would drill just a microscopic amount of powder from a shard, run it through the mass spectrometer, and look for the specific spectral peak that screams hominin, meaning human or human-like.
It's literally like panning for gold in a river of bone shards.
And they struck gold several times. They found a parietal bone fragment, which is the side of the skull. They found a rib. And crucially, they found massive molars.
Let's talk about those teeth. Because even before we had a full face, the teeth gave us this huge hint that these guys were just different.
The teeth were enormous.
Yeah.
Truly functionally massive. If you put a modern human molar next to a Denisovan molar, the human one looks like a baby tooth.
Wow.
And the morphology, the actual shape of the biting surface and the roots, was what we call primitive.
Primitive in what way? Like ape-like?
Not ape-like, but ancient human. It didn't look like a Neanderthal tooth, which have their own specific quirks. And it certainly didn't look like ours. It looked like something much, much older.
Okay.
If you had just found that tooth in isolation, without the cave context, you might think it belonged to Homo erectus or Homo habilis, species that lived over 1 million years ago.
So this was the first physical clue that the Denisovans had really deep roots, that they were holding on to physical traits that other human groups had lost along the way.
Exactly. And that leads us perfectly into section 2.
Rewriting the family tree, the paradigm shift.
This is the heavy stuff.
It is, and I want to stress to you listening right now, what we are about to discuss is the absolute cutting-edge stuff. This is the research dropping in 2024, 2025, 2026, because for a long time, the story we all accepted was pretty simple.
The old model was very tidy. It went like this. There was a common ancestor in Africa. About 600,000 or 700,000 years ago, that ancestor split. One branch stayed in Africa and eventually became us, Homo sapiens.
And the other branch left.
Right, they moved into Eurasia, and then that Eurasian branch split again. The ones that went west into Europe became the Neanderthals, and the ones that went east into Asia became the Denisovans.
So under that old model, Neanderthals and Denisovans were the closest relatives. They were sister taxa, and we were just the distant cousins who showed up later.
That was the consensus for over a decade. But the new data has thrown a massive wrench in those gears. It all centers around a group of Asian fossils called the Homo longiclade.
Often called Dragon Man in the media.
Yes, and a genetic dating technique called tip dating.
Tip dating. Which sounds like terrible advice for a waiter, but explain it for us in this context.
I know, it's a funny term. In phylogenetics, which is the science of building evolutionary family trees, dating the exact moment two lineages split is really hard. Tip dating uses the known geological age of physical fossils.
The tips of the tree branches.
Exactly. It uses those fossil ages along with the genetic differences to calibrate the molecular clock. It helps us determine exactly how fast the DNA is mutating over time.
Okay, so they apply this new tip dating method to these newly analyzed fossils from China, specifically the Yongxian 2 cranium.
Which is a fossil that is about 1 million years old. When they mathematically anchored the evolutionary tree with that specific fossil, it pushed all the split times way, way back.
How far back?
The new results suggest that the Denisovan lineage, which we now think includes this Homo longi group, split from the line leading to Homo sapiens about 1.32 million years ago.
1.32 million. Okay, so they are a very ancient line.
But here is the absolute kicker. The tip dating show that the Neanderthals appear to have split off even earlier than that.
Yeah.
Around 1.38 million years ago.
Wait, let me process that.
Take your time.
If Neanderthals split off first at 1.38 million, that means the ancestors of modern humans and the ancestors of Denisovans stayed together longer before splitting at 1.32 million.
Precisely. It completely flips the triangle.
That is wild.
Instead of Neanderthals and Denisovans being the tight-knit team, it suggests modern humans and Denisovans might actually be the sister taxa, to the exclusion of Neanderthals.
That completely changes the narrative of human evolution. We have always thought of Neanderthals as our closest cousins. This suggests Denisovans might actually hold that title, structurally speaking.
It effectively dismantles the whole search for what anthropologists used to call Ancestor X. You know, that hypothetical single species that gave birth to all three of our groups simultaneously. It suggests a much more complex, staggered series of radiations out of Africa and into Asia over a much longer time frame. And honestly, it gets even more complicated when you look deep inside the Denisovan genome itself. Because they weren't exactly pure.
This is the ghost inside the ghost.
Yes. When geneticists analyzed the Denisovan genome really deeply, looking at the fine details, they found a signal. About 1 to 4% of their DNA didn't come from Neanderthals, and it didn't come from the ancestors of modern humans.
It was incredibly ancient.
A super archaic ghost population. The math says this ghost group diverged from the rest of the human family over 1 million years ago, maybe even 2 million years ago.
So who were they?
The leading theory right now is that this was a population of Asian Homo erectus. Literally maybe the very first humans to ever leave Africa who had just been living in Eurasia, evolving on their own for eons.
Okay, so picture this. You have this super archaic group, let's just call them the Erectus ghost, roaming around Asia. They have been there for over 1 million years. Then the ancestors of the Denisovans show up on the scene. And they don't just wave hello across the valley.
No, they interbred. The genetic data shows they mixed around 700,000 years ago, and then likely again much later. The Denisovans basically acted like a genetic sponge. They absorbed this deeply ancient DNA from Homo erectus.
And because our ancestors later interbred with Denisovans, we picked it up too.
Exactly. It's a cascading chain of gene flow, like a relay race across deep time. Homo erectus passed the genetic baton to the Denisovans, who carried it for hundreds of thousands of years, and then passed it to modern humans when we finally arrived in Asia.
So we are literally carrying bits of DNA from a species that walked out of Africa 2 million years ago, all because the Denisovans acted as the bridge.
It's mind-blowing. It really reinforces this newer concept in anthropology of the braided stream.
Right, we aren't a tree with clean, separate branches anymore.
Exactly. We are a river that separates into different channels, and then those channels weave back together, mixing the water before separating again.
And that river carried some very specific survival traits, which we are going to talk about. But before we get to their legacy, we have to solve the biggest mystery that plagued this field for a decade: the face.
We need to put a face on the ghost.
Section 3, putting a face on the ghost. This is honestly one of my favorite parts of the whole research saga because it sounds exactly like science fiction.
It really does. It comes from an amazing team at the Hebrew University of Jerusalem. So they had the DNA from that original pinky bone. They had the high-quality sequence. But DNA is basically just a list of ingredients.
Right. It says you have flour, sugar, and eggs, but it doesn't tell you exactly what the cake looks like unless you know the recipe by heart.
Unless you look at the notes scribbled in the margins of the recipe.
Okay, explain that.
They use a process called epigenetics, specifically DNA methylation.
We probably need a good analogy for methylation for everyone listening.
Let's use the book analogy again. Think of your DNA sequence as the printed instruction manual for building a human body. It's a huge book. Methylation is like someone going through that printed book with a highlighter and a pack of sticky notes.
Okay.
They physically stick a note on chapter 4 that says, skip this, do not read this chapter. Then they stick a note on chapter 10 saying, read this twice, build a lot of this.
Ah, so it regulates gene expression. It's a chemical tag that tells the body which genes to actually turn on and off during development.
Precisely. It's a methyl group, just a carbon and 3 hydrogens, that physically attaches to the DNA strand and blocks or enhances the cellular machinery. And different patterns of those sticky notes result in completely different physical traits. We know from studying modern humans and animals which genes control the shape of the pelvis or the width of the face or the thickness of the skull.
So the researchers looked at the surviving methylation patterns on the Denisovan DNA.
Right, the chemical degradation of the ancient DNA actually leaves a signature that tells us where those methyl groups used to be. So they mapped the Denisovan sticky notes, and they compared them to the sticky notes of modern humans and Neanderthals.
And they made a physical sketch based just on these chemical sticky notes.
They did. They modeled 56 specific skeletal traits. They said, okay, based on this methylation map, the Denisovan should have a wide pelvis, similar to a Neanderthal. But their skull should be entirely unique.
What exactly did they predict for this skull?
They predicted it would be incredibly broad, laterally widened, not long and football-shaped like a Neanderthal, and not globular and round like ours, just extremely wide. And they predicted a massive, heavy jaw with no protruding chin.
That was a huge, bold move, publishing a predicted, highly specific face of a species you have never actually seen.
It's like describing a suspect to a police sketch artist based only on the sound of their voice.
It was incredibly risky for their careers if they were wrong.
It was. But then the universe provided the physical proof.
Yeah.
Enter Dragon Man.
The Harbin cranium.
Yes. This is a fossil that had basically been hiding in plain sight. It was originally discovered in northeastern China back in the 1930s during the Japanese occupation. A laborer found it, realized it was important, and essentially hid it in an abandoned well to protect it from the war.
Wow.
It stayed down that well for decades and only resurfaced recently when the man told his family about it on his deathbed.
That is a whole movie right there.
And the skull itself is a beast. It is massive, beautifully preserved, and at least 146,000 years old. It is one of the largest Homo skulls ever found anywhere.
So what was the debate?
Well, for a few years after it was brought to scientists, there was a fierce debate. Was it a completely new species? Was it this Homo longi? Or was it our missing Denisovan? The physical morphology perfectly fit that primitive description we talked about earlier.
And in 2025, that debate was finally settled.
Settled definitively. They used proteomics, again, analyzing the ancient proteins in the bone, but more importantly, they managed to extract actual mitochondrial DNA from the dental calculus on the skull's teeth.
Dental calculus, that is just polite scientific speak for plaque, right?
Yes, it is. The calcified gunk on his teeth preserved his DNA better than the bone itself did. And when they sequenced that plaque DNA, it was a match. The Harbin cranium is a Denisovan.
And the moment of truth, did it match the epigenetic prediction from the Hebrew University team?
Was the scientific aha moment of the century? The skull matched the epigenetic model almost perfectly.
That is amazing.
It had that massive laterally wide face, the huge dental sockets for those giant molars, the incredibly broad brain case. We finally, after all those years, had a physical face to put to the ghost.
So when you, the listener, picture a Denisovan in your head now, you shouldn't picture a hunched brute. You should picture a robust, wide-faced, incredibly powerful-looking human.
A human built for absolute endurance.
And that brings us to where they lived. Because another huge myth we need to bust today is that there were just these isolated Siberian cave people.
Right. Denisova Cave is in Siberia. It is cold. It is a harsh tundra environment. But we now know that was just the extreme northern periphery of their range.
The frontier.
Exactly. The Denisovans were masters of extreme environments. Their territory spanned from the freezing Arctic all the way down to the steaming tropics.
Section 4. Let's talk about the roof of the world, Tibet.
This is one of the most hostile environments on planet Earth for a human to live in. Specifically, the Baishia Karst Cave. It sits at an elevation of 3,280 meters. That is well over 10,000 feet above sea level.
At that height, the air is incredibly thin. It's a hypoxic environment. If I went up there today without weeks of acclimatization, I would be gasping for breath just walking. I'd have a splitting headache. I certainly couldn't run down an animal.
You would have severe altitude sickness. And on top of the lack of oxygen, it is freezing cold year-round. And yet, researchers found a Denisovan mandible, a jawbone, right there in the cave. It's called the Yajehe mandible. And more recently, they found a Denisovan rib there, too.
And the dating on those bones is surprisingly old, isn't it?
Very old. The remains date to at least 160,000 years ago.
Wow.
That means Denisovans were living up on the Tibetan plateau way, way before modern humans ever arrived in the region. They weren't just passing through on a hunting trip. They were thriving there for millennia. They were systematically hunting high-altitude prey like yaks and woolly rhinos.
Which heavily implies they had evolved biological adaptations to deal with the lack of oxygen, which we are definitely going to get to in the legacy section. But first, let's jump from the freezing heights of Tibet down to the steamy jungles of Southeast Asia.
Talk about an environmental contrast. Cobra Cave in Laos.
Tamgu how to?
Yes. Researchers found a human molar there, and the dating places it between 164,000 and 131,000 years ago. This is a dense tropical rainforest environment. It's hot, it's humid, it is absolutely full of parasites and totally different predators.
And it wasn't just deep rainforest, right? The chemical analysis of the tooth showed something interesting about exactly what they were eating in Laos.
Right. They did isotopic analysis on the enamel, which tells you what kinds of plants and animals the person was consuming. The isotopes showed that these Denisovans were mostly exploiting the open savanna and the forest edges rather than the deep, dense canopy.
Which is interesting because much later, when modern humans got to that exact same region, we focused heavily on deep forest resources.
It shows that Denisovans were highly adaptable to the tropics, but they still had their preferred ecological niches and hunting strategies.
And we've even found them out on islands, haven't we?
Sort of, yes. We have the Penghu 1 mandible, which was actually dredged up from the bottom of the Taiwan Strait by a fishing boat. That represents a coastal subtropical environment adaptation.
So just to recap, you have Denisovans living in the freezing Siberian tundra, up on the oxygen-starved Tibetan plateau, down in the dense Lao jungle. and out on the Chinese coast.
It is what biologists call supreme ecological plasticity.
I love that term.
We used to think of Neanderthals as the cold weather specialists. We thought of modern humans as the smart generalists who could live anywhere. But Denisovans beat us to it. They were hyper-adaptable. They were the ultimate survivors of the Pleistocene.
And logic dictates that because they lived everywhere, spread across such a massive geographic area, they weren't just one big, homogeneous, happy family.
No, definitely not.
This is Section 5, the populations. And it is crucial for understanding how they eventually interacted with us.
Right. We tend to talk about the Denisovans as if they were a monolith, just one species doing one thing. But the genetic diversity found within the overall Denisovan population was staggering.
Oh, staggering.
The genetic distance between different Denisovan groups was larger than the difference between a modern French person and a modern Japanese person. It was probably larger than the genetic difference between any two modern human populations alive today.
That is incredible. So a Denisovan from the cave in Siberia and a Denisovan hunting in the jungles of Indonesia might barely even recognize each other as the same kind of people.
They were vastly distinct lineages. They had been separated by thousands of miles and hundreds of thousands of years of independent evolution. Geneticists classify the main lineages we know about as D0, D1, and D2.
Let's break those down for the listener. Who exactly are the D0 group?
D is the northern group, the ones we found in Siberia and East Asia. They are the ones closely related to that very first pinky bone from the cave. You could think of them as the classic Denisovan. And importantly, this is the group that likely interbred with the ancestors of modern mainland Asians and Native Americans.
Okay, that tracks. What about D1?
D1 is the southern lineage. They are found genetically in populations from islands Southeast Asia and New Guinea. The DNA clock shows that this D1 lineage split completely from the northern D0 Siberian group about 283,000 years ago.
283,000 years. That is a massive amount of time to be separated.
It is longer than our own species, Homo sapiens, has even existed in our current form.
Wow. Okay, and then there's D2.
D2 is the absolute mind-bender of the bunch. This lineage is found introgressed into modern populations in Oceania and parts of Asia, and the genetic divergence is extreme. They split from the other Denisovans approximately 363,000 years ago.
363,000 years.
They are so genetically distinct from D0 and D1 that some prominent researchers actively argue D2 should be classified as its own entirely separate species.
That is just wild. And the D1 and D2 lineages, you mentioned their DNA is heavily present in modern populations in New Guinea.
Yes, very prominently in Papuan and indigenous Australian genomes today.
Yeah.
And that geographic fact has a massive controversial implication for archaeology.
The Wallace Line.
Yes. To get to the landmass that became New Guinea and Australia, you have to cross the Wallace Line.
For those who haven't taken a biogeography class recently, explain what the Wallace Line is.
It is a deep, invisible biogeographical barrier in Indonesia. It's essentially a deep ocean trench. Even during the peak of the Ice Age, when the glaciers were huge and global sea levels dropped by hundreds of feet, this trench was so deep that there was never a land bridge connecting Asia to Australia.
Never?
Never. It perfectly separates the fauna of Asia from the fauna of Australasia. That's why you have tigers, elephants, and monkeys on the Asian side, but you only have marsupials, kangaroos, and koalas on the Australian side.
Because the tigers couldn't swim across.
Exactly. Elephants couldn't cross it either. Getting across the Wallace Line requires crossing deep, treacherous open water with incredibly strong, sweeping currents.
But the Denisovans did it.
The genetics strongly imply that they must have. If their DNA is deeply embedded in the indigenous populations of New Guinea and Australia, and those modern human populations have been isolated there for 50,000 years, the Denisovans had to have been there first to interbreed with them when they arrived.
Which implies boats.
It absolutely implies maritime capability. Maybe they built bamboo rafts. Maybe they had simple canoes. We haven't found a petrified Denisovan boat yet, obviously. Wood doesn't survive that long. But the genetic maps don't lie. They got a breeding population across deep open water.
That totally challenges everything we thought we knew about archaic human intelligence and technology.
It does. We usually draw a hard line and say boats equal modern human cognition. But Denisovans might have been open water sailors, hundreds of thousands of years before we ever dipped a toe in the ocean.
And that segues so perfectly into Section 6, their culture, their tools, and their trade. Because if they had boats, or at least rafts, what else did they have? We used to think of archaic humans as just scraping by, banging rocks together. But the artifacts tell a totally different story.
It is what archaeologists call the initial Upper Paleolithic shock. When we look at the dirt layers in Denisova Cave that date to around 45,000 to 50,000 years ago, we find things that look like they belong in a modern art gallery or a high-end tailor shop.
Okay, tell me about the ostrich eggshells.
This is one of the most stunning finds. They found beads, beautifully, delicately crafted personal ornaments made from pieces of ostrich eggshell.
Just for decoration.
Yes, for adornment. And they weren't just roughly chipped. They were drilled with precision stone drills. High-speed, microlithic drills.
High-speed drills, like a Dremel tool.
Hand-operated, of course, using a bow or a pump mechanism.
Yeah.
But yes, rotary motion. The holes in these fragile beads are tiny and perfectly centered. If you tried to punch a hole in an eggshell with a clumsy, heavy stone tool, you would just shatter it into dust.
Right.
It requires intense dexterity, patience, and highly specific, specialized tool-making skills.
But wait, a more basic question here. Are there even ostriches in Siberia?
No. And that is exactly the point. Ostriches absolutely did not live in the freezing Altai Mountains. The nearest known ostrich populations at that time were way out in Mongolia or the Transbaikal region.
So they imported the raw materials.
Yes. Either they personally traveled huge distances to gather these shells, or, much more likely, they had established trade networks. They were interacting and trading with other groups of hominins.
Like, I'll give you this premium mammoth ivory if you give me those eggshells.
Exactly. And trade implies so much more than just swapping items. It implies complex communication. It implies forward planning. It implies a shared system of value. It implies a complex economy operating 50,000 years ago.
And then there is the needle.
Oh, the eyed needle. It is a masterpiece. It's made of bird bone, and it is dated to 50,000 years ago. It is currently the oldest eyed needle ever found in the world.
Let's pause on why an eyed needle is such a massive leap in technology. It's not just a sharp, pointy stick.
Right, and awl, which is just a sharp stick made of bone or stone, is used for punching holes in thick leather, so you can thread a heavy leather thong through it and tie it together.
Like tying a tarp.
Exactly. But an eyed needle, with a tiny hole drilled through the back end, is for pulling thin thread. It is for sewing.
It's for tailored clothing.
Precisely. You do not spend the hours required to carve a delicate bone needle unless you are sewing tight-fitting, multi-layered garments. We're talking fitted trousers, parkas with hoods, insulated gloves.
Which makes perfect sense because they lived in Siberia during an ice age. You cannot survive a Siberian winter wrapped in a loose, drafty mammoth skin toga.
No, you would freeze to death in a day. You need fitted tailoring to trap a layer of warm air against the skin.
So let's look at the picture we've painted so far. We have a people who can survive in hypoxic mountains, who can potentially navigate open oceans, who engage in long-distance trade, and who sew tailored clothing. They are, for all intents and purposes, culturally indistinguishable from us.
Cognitively and culturally, the gap between them and us is vanishingly small. They were not primitive cavemen. They were just different humans.
And clearly our ancestors found them very attractive, because we didn't just trade beads with them, we slept with them.
We certainly did, and so did the Neanderthals.
This brings us to Section 7, Love, War, and Hybrids. Let's talk about the fossil known as Denisova 11.
Deni is honestly the holy grail of ancient DNA. She's a tiny bone fragment discovered back in 2012. The bone is about 90,000 years old. And when the lab sequenced her genome, the results were so unbelievably clean and perfectly split that the researchers genuinely thought they had made a mistake or contaminated the sample.
What exactly did they find?
They found that her mother was a 100% Neanderthal,
Okay.
and her father was a Denisovan.
A first-generation hybrid, a literal 50-50 biological mix of two completely different human lineages.
Yes, and it is incredibly rare to find a first-generation hybrid of anything in the fossil record. Think about the astronomical odds here.
Right.
We have found maybe a dozen distinct Denisovan individuals total in all of history. And one of those random dozen just happens to be the direct first-generation child of a mixed species couple.
That heavily implies it wasn't a rare event at all.
Exactly. If it were a 1 in a million fluke event, we would never have found it in such a microscopic sample size.
Yeah.
The sheer statistical fact that we found Deni suggests that whenever these groups met out on the landscape, they mixed. They didn't view each other as terrifying alien monsters. They recognized each other as people.
And the father, the Denisovan dad, he wasn't exactly pure either, was he?
No, he wasn't. When they looked at his specific chromosomes, they found that deep in his family tree, hundreds of generations back, there was ancient Neanderthal admixture too.
Wow.
So these distinct groups were constantly continually swapping genes back and forth for millennia. It wasn't just a one-time Ice Age fling. It was a standard feature of Pleistocene life.
But it wasn't all romance and easy blending. There is evidence baked right into our own modern genome that this hybridization didn't always work out biologically.
Right. It wasn't completely smooth sailing. This brings in a biological concept called Haldane's rule.
Explain Haldane's rule for us.
Haldane's rule states that when two different species or highly divergent subspecies interbreed, if one sex of the resulting offspring is sterile or non-viable, it is almost always the heterogametic sex. In mammals, like humans, that means the male, who has an X and a Y chromosome.
Because the male has two different sex chromosomes.
Right. If there are genetic incompatibilities, mutations on the X chromosome that clash with the other parent's genes, the female offspring, who is XX, has a backup copy of the X chromosome to compensate. She might be perfectly fine and fertile.
But the male doesn't have a backup. He just has the Y. So if the X is incompatible, he's in trouble.
Precisely. If the genes clash, he is likely sterile.
And we actually see the ghost of this rule in the modern genome.
We absolutely do.
Yeah.
We see what geneticists call integration deserts. If you look at the DNA of modern humans today, there are huge chunks of our X chromosome that have completely zero Denisovan or Neanderthal DNA. It was actively weeded out by evolution.
Meaning those genes were detrimental.
Yes. And specifically, genes related to modern male fertility. They contain no Denisovan DNA either.
So the male hybrids back then were likely sterile.
It's highly probable. The female hybrids, like Denny, were fertile and could successfully pass the mixed genes back into the wider population. But the boys, they likely couldn't have kids of their own.
That is fascinating.
It suggests we were right on the absolute brink. The species were still compatible enough to mate, but barely. Give it another few 100,000 years of separation, and we might have been completely biologically incompatible.
But enough of that DNA did survive the bottleneck. And this brings us to Section 8, the legacy. This is the ultimate so what of the episode. Why does this matter to you, the listener, sitting in traffic or folding laundry right now? It matters because the Denisovans aren't fully extinct. They are literally inside you.
This is the concept of adaptive introgression. I like to think of it as an evolutionary software update.
I love that analogy. Unpack the software update for us.
Okay, so when modern Homo sapiens migrated out of Africa, about 60,000 years ago, we were immunologically and biologically naive to the rest of the world. We were tropical African animals. We did not know the specific pathogens of Eurasia. We were not biologically adapted to extreme cold or to high altitudes.
But the Denisovans had been living there for hundreds of thousands of years. They had already done all the hard evolutionary homework.
Exactly. They had suffered, mutated, and adapted. And by interbreeding with them, our ancestors essentially borrowed their genetic cheat codes. We took 400,000 years of grueling evolutionary R&D and just downloaded it into our own modern human genome in a few generations.
Let's give the listeners the big examples, the biological superpowers we inherited.
The most famous, without a doubt, is the EPAS1 gene. This is the superathlete gene found heavily in modern Tibetan populations.
What exactly does EPS1 do in the body?
Normally, if a person from sea level goes to extreme high altitude, the body panics. It senses the low oxygen in the thin air, and it reacts by pumping out a massive excess of red blood cells to try and capture more oxygen.
Which sounds helpful, but it's not.
No, it leads to a condition called polycythemia. Your blood gets dangerously thick. It's like your heart is trying to pump sludge through your veins. It causes severe heart problems, blood clots, strokes, and it is extremely dangerous for pregnancies.
But Tibetans who live up there don't get that thick blood.
Because they have the Denisovan variant of the EPAS1 gene. This variant basically acts like a dampener. It tells the body, relax, do not overreact, we are fine. It keeps hemoglobin and red blood cell levels stable, even in severely thin air.
And that specific variant came straight from the D0 Denisovan lineage.
Directly from them. Researchers found the exact same highly unique gene sequence in the ancient Denisovan DNA from the cave. Without that specific Denisovan gene update, permanently settling the Tibetan plateau would have been nearly biologically impossible for modern humans.
We literally stand on the roof of the world thanks to a ghost.
We do.
That is incredible. What about immunity? Because I know that's a huge part of the legacy.
Yes, There's a gene called TNFAIP3. It codes for the A20 protein in our immune system. The Denisovan version of this gene basically supercharges your inflammatory immune response. It's an immune switch with a hair trigger.
Which was a good thing back in the Pleistocene.
It was essential for survival. When our ancestors entered the dense tropical jungles of Southeast Asia and Oceania, the pathogens there were aggressive. Brand new viruses, nasty bacteria, parasites. You needed a hyper-aggressive hair-trigger immune system just to survive childhood. The Denisovan gene gave our ancestors that protection.
But there's a trade-off today, isn't there?
There always is in evolution. Today, in the modern world, most of us live in relatively sterile, sanitized environments. We aren't fighting off novel jungle parasites every single day. But that genetic hair trigger is still sitting there in our DNA, waiting to fire.
So it gets bored and starts attacking us.
Exactly. That specific Denisovan immune variant is now strongly linked to modern autoimmune diseases, things like rheumatoid arthritis, lupus, and inflammatory bowel disease.
So if you are listening to this and you suffer from arthritis, it might literally be a direct legacy of a Denisovan ancestor trying to keep your great-great-grandfather alive in a prehistoric jungle.
It is the lingering cost of ancient survival. It saved your ancestors 40,000 years ago, even if it causes your joints to swell today.
Nature is brutal. And there's one about cold adaptation and body fat, right?
Yes, the TBX15 and WARS2 gene complex. These genes help the body generate heat from brown fat, which is a specialized type of lipid metabolism. We find this Denisovan sequence introgressed very heavily in modern Inuit populations and indigenous Americans. It actively helped them survive the brutal cold of the Arctic during their migrations.
And there's a really new one that was just identified recently, too, MUC19.
Yes, MUC19. It's a gene that creates a thicker, more robust mucus barrier in the mucosal membranes of the gut and the lungs. It is found frequently in indigenous populations in the Americas. It most likely provided enhanced protection against severe respiratory and gastrointestinal pathogens during that long, grueling migration across the Bering Strait land bridge.
It is just amazing to step back and look at this. We, Homo sapiens, we like to think of ourselves as the ultimate conquerors of the world, the wise man. We think our big brains did it all on our own.
We write the history books to make ourselves the sole heroes.
Right, but the truth is, we absolutely could not have populated the globe without their help. We stood on the shoulders of giants, or at least the wide shoulders of Denisovans.
We are a composite species. Our incredible global success is directly built on the deep genetic diversity that we absorbed from these other older groups. We are a biological mosaic.
So let's wrap this mystery up. We started with a single pinky bone, just one tiny overlooked fragment in a freezing cave in Siberia. And from that one bone, scientists have unraveled a massive dynamic population that lived for over 1 million years.
They spanned an entire continent.
They sailed across ocean trenches, they traded luxury goods, they sewed tailored winter clothing, and eventually they merged with us.
It is the ultimate scientific detective story. And the craziest part is that the map of humanity is only getting more crowded. Think about it. If the D2 lineage was so genetically different, and we know they were living down in Oceania 40,000 years ago.
What else is out there?
Exactly. What else did they build?
That is the provocative thought I want to leave everyone with today. If there was a highly divergent, deeply ancient human species living in Oceania tens of thousands of years ago, a species clearly capable of crossing the Wallace Line, what did they leave behind? What ruins or tools or carved bones are currently sitting buried under a thick layer of jungle mud in Papua New Guinea, just waiting for a zoom scanner to find them?
We have barely even scratched the surface.
Yeah.
The ghosts in our genes are speaking, and I promise you, they have a lot more to say.
If you enjoyed this deep dive into the ghosts of our ancient past, please make sure to follow and subscribe so you don't miss the next scientific mystery we unravel. We will see you next time on The Deep Dive.
Keep looking deeper.